just going to lecture. They haven't told me if they can't hear me. Like Tough Bounce, watch it on YouTube. Zoom in just a little bit here. Alright. This is awkward, but you know, we're just gonna go with it. Okay, well, good morning. Today we're continuing on with Chapter 3, looking at the building blocks of life. And so far we've covered carbs, and we started with nucleic acids. We didn't finish, so I'm just going to go back a couple slides here. So we started to talk about the structure of DNA and RNA and the differences between the two. They have different sugars. Make sure you know differences between RNA and DNA. I'm pretty sure it's an exam question. So they have different sugars, the ribose versus the deoxyribose. RNA is single-stranded. DNA is double-stranded. The base pairing is a little bit different. The base pairs, I should say, are different. RNA, A, U, C, G, and DNA, A, T, C, and G. So know at least 3 differences between these molecules. You can even go a step further and say RNA is unstable and DNA is stable. James, can you hear me? James? See, this is why hi. Okay, flipping on the lights. Gosh darn it. Okay, we're good. Give me a thumbs up. All right.
Why do they mute?
I don't know. They're the only ones. They really shouldn't. But they can hear me, so. All right. So no differences between RNA and DNA. Okay. So we didn't finish nucleotides. We talked about the prebiotic soup, how some inorganic molecules formed early in life's history and how they could have developed some molecules that are necessary for life. Talked about the origin of pyrimidines, how that's challenging. So there's some holes to this hypothesis. And this is where we're at. So we're going to focus in on DNA and how it carries the genetic code today. So we have sequences of nucleotides that carry information about which amino acids should be put together. It's kind of like a recipe for which proteins are made. So proteins are made from amino acids. So we'll talk about proteins, I think, in the next section here, but sequences code for amino acids. Shape of DNA is double helix, 2 strands held together by hydrogen bonds. Remember yesterday, I showed you that diagram, and we talked about the 3 hydrogen bonds, the 2 hydrogen bonds, how hydrogen bonds is not a sharing of hydrogen or any electrons. It's really just a force of attraction between different molecules, so it's outside the molecules. Base pairing rules are complementary. A pairs with T, C pairs with G. So our DNA is located in the nucleus, double helix, and we talked about the central DNA dogma, how DNA gets transcribed into mRNA, which is single-stranded, and there is complementary base pairing here. G pairs with C, but instead of T on RNA, they have a U. So T is replaced by U, U pairs with A. We have the single-stranded molecule that leaves the nucleus and then will get translated by a ribosome and will form a polypeptide, basically a sequence of amino acids stringed together like pearls on a string. And then when it's done assembling the amino acids, the protein or the string of amino acids will fold into a protein, and that protein will do something for the cell. So RNA is a transcript of the DNA strand. So what I mean by transcript is that it's basically reading the DNA and getting that information, kind of decoding it. Similar to DNA, 2 major differences. I just said contains ribose sugar, not deoxyribose, and then also utilizes uracil in place of thymine. Okay, single-stranded molecule to specify the sequence of the amino acids. So capital A, amino acids. So again, one more picture comparing RNA to DNA, single strands versus double strand, different sugars, ribonucleic, deoxyribonucleic, and different bases, CGAU, and then over here, CGAT. If I were to ask you guys the question, what is a similarity between RNA and DNA, what would you guys say? Sorry, I'm going to put you three on the spot.
They have the what are those things called? Cytosine.
Yep, so they have cytosine, guanine, and adenine the same. That's definitely one similarity. What's another similarity? Okay, Herman Norcross, your turn. Brooke did her part. Say that again. All right, Brooke nailed it again. I didn't hear you guys over there, but they both have a sugar phosphate backbone. All right. All right, there are some other nucleotides that are vital components of energy. So I know when we were talking about phosphate, because phosphate is found in a nucleotide, I mentioned 3 phosphates paired with an adenine makes ATP. And ATP is the molecule that our body uses for energy. I read a fun fact about ATP. Actually, a couple years ago, someone asked the question, how much ATP does your body have? And I was like, whoa, you know what? I'm not sure. Because it's obviously, every single cell uses ATP to do something. So I decided to look it up. And they said that you only have like 50 grams of ATP in your body. Because what happens is when you make ATP, I'm just going to use the A for the adenine and then the letter P for the 3. Well, what happens is you cleave off this last phosphate, and now instead of ATP, you get ADP. And then when you want to make energy, then they add another phosphate to it to go back to ATP. So ATP churns or is converted into ADP, and then it gets converted back to ATP, and it has to do this very quickly. And so we'll talk about those processes later on this semester.
What's ADP? Is that
adenine diphosphate, because it only has 2. Yep. So ATP is this energy currency of cells that is constantly being used, converted to ADP, back to ATP. So your body has to, it burns it really quickly, and then it has to reform it again. So it drives all energetically unfavorable chemical reactions, moves things across your cell membranes. It's used for muscle contractions and all that. Another important molecule that's associated with nucleotides is NAD+. And it's got that adenine in it, which is a nucleotide, nicotinamide adenine dinucleotide. NAD+ and FAD both have adenine in it, and both of these guys are considered as electron carriers. In our chapter of photosynthesis and cellular respiration, we will talk a lot about electron carriers. Basically, they pick up electrons and they move it somewhere else. So very important in the process of creating energy or making food. Okay, so that does it for nucleotides. And now we're going to switch gears and talk about proteins. And so we're going to look at the structure of an amino acid and what makes the amino acid different from another amino acid, which is the R group. And then we're going to look at 6 levels of protein structure and the forces that help create that protein. And then also at the very end of this little section, talk about chaperones and how they are involved in protein folding and unfolding. This section is really important for your enzyme lab, which is coming up. So obviously, I don't think I'm going to get through everything I want to get through today. So our lab will be sometime next week, and then after the lab will be the exam. So originally, I had planned to do the lab tomorrow with an exam on Monday, but that's not the case here. So just know that you have an exam coming up next week, and we also have a lab next week. So proteins, main structure and function here, wide range. I mean, Proteins do a lot of the work inside of our cells. They help carry out chemical reactions. So I have 7 categories of functions. And the first one is an enzymic catalyst, which means that they help chemical reactions occur. Enzyme catalysts are basically enzymes. Okay, they're these 3D globular proteins, and what they do is they fit snugly. Wow, they fit pretty snug with specific molecules. So here's my enzyme, and here are my reactants, and the reactants bind to the enzyme. A chemical reaction occurs, and now I've created new products. Now this diagram is actually looking at a disaccharide of glucose and fructose, and an enzyme that is very specific to the shape of this molecule. As you can see, it fits really nicely in there. Undergoes a chemical reaction, splits the 2 sugars back into monosaccharides. That's just one of the many enzymes inside your body. So you have enzymes in your saliva, salivary amylase. You take a saltine cracker, put it in your mouth, and you just keep chewing. Keep chewing. It's going to get really disgusting. Okay, it's kind of like, hey, do you like fish food? That kind of joke, you know? You just keep chewing. And eventually what's going to happen is you're going to start to taste a little sweetness. And that's your salivary amylase going to work, breaking down complex carbs into simple sugars. So enzymes, they help create chemical reactions or start chemical reactions. Now, the thing I want to emphasize with enzymes is that they are not in the chemical equation. You know, like I could write out a chemical equation for this, how glucose or this disaccharide, whatever the name of it is. Now, this would be sucrose actually, because glucose plus fructose is sucrose. So sucrose breaks down, decomposes into glucose and fructose, but you would never see the enzyme involved in that. So these enzymes start the chemical reaction, or allow it to take place, but they're not on the reactant or the product side. And once they do the chemical reaction, split it into new products or whatever they're supposed to do, they just go and find another sucrose molecule and do it again. So they are reusable, and you will see that in your lab. You'll have an example where it's like, is this enzyme reusable? And you'll do a separate set of experiment, and you'll say yes or no. So, okay, second function of proteins that they can serve in is defense. There are proteins that, their main job is to recognize specific microbes as well as maybe cancer cells. So like inside of our immune system, we have lots of proteins that go around and they bind to cells and they say, hey, are you a part of me, like the organism, or are you a foreign material? And if you are a foreign material, well, now it's going to start a signal transduction pathway saying, hey, this is foreign material, we need to get rid of it. It will recruit other cells to help carry out that task. So you see it a lot in endocrine and immune systems. I also want to mention venom. Okay, venom work on receptors on the outside of certain cell membranes. So like here we have a scorpion, and there's venom obviously in the stinger of the tail. And in this case, this venom is called chlorotoxin protein. So the venom contains proteins that block chloride ions from entering inside the cell. And as a result, the cell will start to dysfunction. It's not going to work properly anymore. Might lead to cell death or just an infunction of, a malfunction. That's the word I'm trying to find here. A malfunction of the cell resulting maybe in cell death or even death of the organism. Because especially if this cell is part of like your heart or your diaphragm, which allows you to breathe, and all of a sudden those muscles, those cells aren't working properly anymore, that's bad news for you. Okay? So defense. And I think on the next slide I have a picture of snake venom as well with a table that gives you site-specific cells that it targets. Third function that proteins can serve is they help transport small molecules and ions. So hemoglobin transports oxygen in your blood. It's hemoglobin is a protein. Myoglobin is another protein. Carries oxygen in your muscles. Transferrin helps transfer iron in blood. So proteins can help move things inside and outside the cell. Even this is a protein. Okay, it allows chloride ions to travel through. So channels are proteins. And so we'll talk more about cell structure later on this semester and how there are certain proteins embedded into the cell membrane. that give it, characteristics and properties. So here's a picture of snake venom. And the thing I really just want to work on is the salivary gland up there. But it has, there's different snake toxins, and it gives you the effects of neurotoxin. This one, disruption of nerve impulses causing heart and respiratory failure, atom tissue decay. These guys work on acute low blood pressure, anaphylactic shock, paralysis of the peripheral smooth muscle. Okay, so paralysis means that it just like seizes up, and then peripheral smooth muscle. Smooth muscles are like examples of that would be muscles that you don't really control; they just happen automatically. So like your esophagus, your stomach, your intestines. So that's smooth muscle. Muscle decay and neurotoxicity. Again, acute low blood pressure, shock, destruction of blood clotting factors, which means that maybe you could bleed to death if you had several bites. Constriction of intestinal muscles resulting in cramping, increased perception of pain, and then, so lots of different toxins with their effects, and they're acting on, usually, proteins on these cell surfaces. Okay, fourth function could be support. Protein fibers provide structural support, keratin in hair and nails, fibrin in blood clots, collagen is a protein. It's found in your skin, ligaments, tendons, and bones. It is the most abundant protein in your body. Fifth way that proteins can help you out is motion. Our muscles use two different types of protein filaments, actin and myosin, creating cross bridges. So here we have myosin filament, and then in blue here, the actin filament. And what happens is that these myosin heads kind of cling to the actin filament, and they just like grip it, and they twist and move. And as they move, the muscles contract. So you can see here that the myosin heads are, they've climbed far along the actin filament. And then when they want to relax, they let go, and now your muscle is relaxed. So we have a lot of contractile proteins that involve involuntary movement. And then the last two is regulation, hormones as intercellular messengers. They help turn on and off genes during development, and can receive information saying, oh, hey, this is happening. Maybe you're doing a flight or fight response, so hormone levels kind of change there. Last one, storage, calcium and iron stored by binding as ions to storage proteins. So there are proteins that store calcium and iron inside of you. Okay. So lots of different functions. So let's take a look at the building blocks of a protein, the building blocks of a protein, basically. Amino acids. There are 20 different amino acids out there, and so the structure of amino acids are similar except for one difference. So here's the structure of amino acid. You have an amino group, hence the prefix, amino. You have hydrogen. You have a carboxyl functional group here, and this is the only thing that's different, the R group. This is what makes, the difference between a, let's see, I can't even think of an amino acid right now, aspartic acid versus glutamine, okay? So it's very similar to our nucleotides, like how we said a nucleotide has a phosphate binded to a sugar. So like these two are the same, but the difference that makes it whether or not it's an adenine or guanine is the nitrogen base. Well, same thing with an amino acid. They have an amino, a hydrogen, carboxyl. That's the same. This is the only thing that's different that makes. Do you have a question, Herman Norcross? Nope. Okay. So the functional side group, or as it's labeled, R group. So the unique character of that amino acid is determined by R. It could be chiral, could exist as two enantiomeric forms, a D or an L. So there's that chiral, left-handed, right-handedness. L amino acids are found in proteins. D amino acids are rare. And then R group determines the unique chemistry of that amino acid, whether or not it will be polar or non-polar.
So these are the groups to classify them?
Nope, that's going to be on the next. Is it saying like what are the 5 classes?
Yeah.
That's the next
5
slide.
groups to classify.
Oh, wait, hold on. Let me take a look at it real quick before I say the wrong thing. Yep, this is this slide right here. Okay, so there are 20 amino acids, and we group them into 5 classes based on that R group variant. And so the first one is nonpolar amino acid, and the R group is the CH2 or a CH3, and an example of a nonpolar amino acid would be leucine. So nonpolar means that it will not react with water, like it will be hydrophobic in water. Polar uncharged amino acid, the R group is an O or an OH, tyrosine. You can have charged amino acids, where the R group will be an acid or a base that can ionize, so glutamic acids. Usually you see the word acid at the end of that. Aromatic means that it has a ring. So I talked yesterday about the double ring. The ring could be that, or it could be, it's just a ring shape of carbon ring. Alternating single bonds or double bonds. Aromatic rings are usually nonpolar. So yeah, we'll just say ring amino acids. And then the last one's kind of this catch-all. If it's not these 4, we're just going to call it a special function. So methionine is one. Proline is another. Cytosine links chains together, so they have characteristics or properties that they do. So methionine is always used in the first amino acid. When you make a protein, that's always the first amino acid. It says proline causes kinks in changes, and so we will talk about proline later on. And then cysteine links chains together. So yeah. Each amino acid affects the shape of a protein differently because you're stringing along polar, nonpolar, charged, aromatic. And then when you got your string of proteins, then they're going to start interacting with each other. Oh, geez, this nonpolar, we'll just say leucine right next to threonine, is going to try to repel each other. They're trying to get away from each other because they don't like each other. So here's a diagram showing the 20 amino acids and how they're grouped. As you know, are they charged, uncharged, special cases, and then hydrophobic rings. And so you can see the aromatics here, which means that they will be nonpolar properties. So I know I said there are 5 classes. Well, they group the aromatic rings in with the nonpolar because that's a property of them. So 1, 2, 3, 4, 5, and then this obviously belongs to this. So proteins are made up of chains of amino acids, and the amino acids are held together by peptide bonds. It's a covalent bond. So covalent means what with electrons? Are they being transferred or are they being shared?
Is that a question?
Yep.
Okay, ask again.
Okay. Covalent bonds are electrons being transferred or are they being shared? They are being shared. Okay. So the covalent bond that is, peptide bonds are covalent bonds. What happens is we have that dehydration reaction that forms the covalent bond. So that means water will form. Okay, dehydration is the same thing as condensation. So when you form bonds, water will form. If you want to break bonds, it's like you're adding water. This bond is not free to rotate around the N-C linkage, and so as a result, sometimes it acts like a partial double bond character, where it's a little bit more rigid, but it's also going to give it some structural character. But when you string together a bunch of amino acids using peptide bonds, eventually you're going to get that polypeptide. So many peptide, which means many peptide bonds. Okay. So a protein composed of one or more large unbranched chains linked by peptide bonds. And I can't stress this enough. There are 20 common amino acids. There's a reason why that information is being repeated. Okay. So here's how a peptide bond forms. I have an amino acid here, an amino acid here, and they have the R group. They didn't, we don't know what type of amino acid it is, but it's just saying, hey, here's my amino group. There's the H and there's the carboxyl. Well, what happens is that the link happens between the O and the H, these two H's I should say on the amine side, and you form your water. Now we have a peptide linkage, and this bond is very rigid. Okay, so it almost acts like a double bond, but it's not. And yeah. Okay, we're going to talk about protein structure here, okay, because the shape of a protein is really important for what kind of job it will carry out. So I know I have already mentioned this story before, and I do have a picture of where I mutated our protein on the next slide here. So this is actually from my paper that I had to write for the class. So I was looking at circadian rhythms, specifically the interaction between cry and PP5. And PP5 is the gummy bear-shaped protein. And our professor hypothesized that maybe the limbs of the gummy bear, fit really nicely with the protein, kind of like how an enzyme fits with its reactants and undergo a chemical reaction. So we thought that, it somehow binds to PP5 to start the circadian rhythm. So cry is a pacemaker. It's activated by light. So when your body senses light, cry gets produced and binds to PP5 to start this cascading effect of your biological clock. So then PP5 goes on to interact with another protein, in this case CKIE, and then it goes on inside the cell. This is supposed to represent the nucleus, and this is supposed to represent genes that code for proteins that your body may need. And it unblocks this, and you can make proteins with it. So we were looking at the interaction of these molecules inside a cell. So then each of us had to mutate a part of the gummy bear. So we did selective mutation of residues on mCry1. So basically this little M, whenever you see a little M in front of something, that means it was mutated. So yes, we actually got to alter the amino acid sequence of this protein and thus change the charge. So here's a picture, a computer model showing the electrostatic potential where red is negative and blue is positive. And I mutated aspartamine 136. So this is the amino acid, and it was the 136th amino acid in the sequence. And I don't remember how big this protein was. It was a couple hundred. So I mutated the 136th amino acid, which was aspartamine, on mCry1. So this is before the mutation. This is after the mutation. As you can see, we kind of lost a lot of the negative properties or characteristics of it. So I was like, all right, let's just see now if cry will still bind to it. If cry doesn't bind to it, then I picked the right mutation. My paper would go on. Obviously, my professor would take credit for it, and then she'd be, go on to be famous or whatever. So mutated it. I switched it to an alanine, producing a neutral charge, as you can see down here. But in my experiment, I failed. mCry still bound to PP5. Okay. Nonpolar amino acids are found internally with one another, and polar charged amino acids are. Yes.
What are the guided, Chapter 3 guided note taking you guys do?
When I'm done with this, probably the same day as the lab.
Okay.
Which we haven't done. So you got the weekend, basically.
Okay, thank you.
Yep. So nonpolar amino acids usually hang out with each other. So here I have some helices of amino acids, okay? And this indicates polar, and red indicates nonpolar. So the polar will interact with each other, and the nonpolar will I don't want to hang out with positive, like polar molecules, because they don't like each other. So they will either flip or invert. Here's carbon chain showing nonpolar R groups, two polar R groups, and so they will avoid each other because they don't mix. Now, if these were all polar, then I'm sure that they would flip and they would all interact with each other, maybe forming a ring. Who knows? But they will avoid and repel each other.
What's that noise?
It's raining right now.
Yeah, I was gonna say, it sounds like it's raining.
Yeah, it sounds pretty hard right now. So proteins do have 4 levels of structure, and so the first level of structure is the primary structure. This is the easiest, It's your amino acid sequence. Okay? Now, any of the 20 different amino acids can appear in any position. So if I was looking at a 100 sequence amino acid protein, where it was just made of 100 amino acids, the possibilities of the combination of amino acids is really great. It would be 20 to the 100, because an alanine can be paired with another alanine, so you can double them up. It does, just because you have an alanine doesn't mean that it can't be an alanine. So very diverse. That's why proteins are just a wide range of them that do different things. Hemoglobin, picture down here, found in your red blood cell, helps carry oxygen, is actually made up of big globular proteins, 2 alpha globulin peptides and 2 beta globulin peptides. And if you alter the sequence, you can change the protein. This is what happens with sickle cell anemia. You change like a valine or to a glutamic acid or vice versa. I don't remember offhand. But as a result, instead of being this nice little donut-shaped red blood cell, it turns into like a crescent moon. And it's not going to carry oxygen as effectively. And it's all because of one mutation in the amino acid sequence. So, yeah, you change the amino acid, you could change the shape and the structure of the resulting protein. So a picture showing the primary structure, where it's just basically pearls on a string, and they can be in any order. That's primary structure, amino acid sequence. Secondary structure involves hydrogen bonding. Okay, so H bonds can be either with water or with other peptide groups. And remember, with a hydrogen bond, it's not a transfer of electrons, it's a force of attraction. There are 2 forms of secondary structure, alpha helix and beta sheets. Alpha helix, think the shape of DNA. The peptide groups are interacting with one another, and as a result, they will coil into a spiral that's very similar to DNA. A beta sheet, what happens is that it's kind of flat, but it's rigid. It's kind of like making a paper fan. So when you make a paper fan, that's what a beta sheet kind of looks like. It can be parallel, anti-parallel. We'll talk about aquaporins later on this year, but aquaporins use beta sheets. Hemoglobin uses both alpha and beta. So here's a picture showing secondary structure, where here we have the helix, cylindrical. Okay, here's the beta sheet, and you can see that it looks like a paper fan. So alpha helix, beta sheet, and they are the results from hydrogen bonds. So secondary structures, hydrogen bonds. They can be alpha or beta. The third structure in proteins is tertiary structure. This is where the proteins start to fold and kink based on, maybe nonpolar, polar van der Waals interaction. So this is supposed to represent the amino acid sequence, but sometimes we get these side chains that start to interact with each other. Hydrogen bonds, hydrophobic interactions. Okay, we're going to fold and almost like cut ourselves off from polar molecules. Disulfide bridges. The disulfide bonds are responsible for making your hair curly. So when people get a perm, they're actually adding the disulfide bonds to their hair to make it curly. Ionic bonds. So just like additional folds and kinks, giving that protein some more characteristics. Okay, see ya tomorrow. So tertiary, continued. This is the list of the forces involved in tertiary structure. I mentioned H bonds. So yes, H bonds are responsible for secondary structure that, they're solely secondary, but they can occur in the tertiary portion. Electrostatic attraction, opposites attract, positive and negative. Hydrophobic exclusion, this is where the nonpolar R groups hang out and try to cut themselves off with polar regions. And then covalent bonds, like the disulfide bonds I showed you on the previous slide, resulting in curliness of hair. Strength of stability is influenced by how well the interior fits together. So here I have a hydrogen bond, And hydrogen bonds, remember, is a force of attraction. So a hydrogen bond next to an ion bond might make this portion of the protein a bit stronger. Notice down here we have some hydrophobic interactions, specifically with van der Waals. So depending on where they're at, you might take it a step further and have your hydrophobic portions participate in van der Waals, which we talked about with geckos. Okay, the final structure of protein is the quaternary structure, and these are the subunit arrangements. So after your beta sheet or your alpha helix is done and it folds and does all the kinks, if it binds to like another subunit, so I like how in this diagram they color code this one orange. So the orange interacts with other subunits, in this case purple, green, and yellow, to form your functional protein, if you will, kind of like the final product, the final stage of a protein, quaternary structure. So this diagram, I think, just pieces it all together really nicely. Here's my sequence of amino acids, primary structure. In this case, I'm looking at an alpha helix, secondary structure, hydrogen bonds. Tertiary, I start to get more folds and kinks. We're starting to see other interactions, ionic bonds, covalent bonds, nonpolar hydrophobic interactions, and then when it connects or binds with some other assembled subunits, you get that final protein. Now, because proteins are so diverse, okay, there's a lot of proteins out there, scientists kind of use some clues to figure out what it does. So at first, to determine the sequence of amino acid, it was very slow and difficult. Now it's pretty fast. We have great technology. DNA is directly related to the amino acid sequence in proteins. We know that. But we can also look at something called motifs and domains to figure out what the protein may be involved with. So let's look at what the word motif means. It's actually a supersecondary structure. So when we first discovered the shape of a protein, we noticed similarities between other proteins that we did not know. So that's what dissimilar proteins mean. Okay, we know hemoglobin, it carries oxygen. Hey, here's a protein, we don't know what it does, but it has similar properties or shapes of hemoglobin. Maybe it does the same thing, but just in a slightly different way. So that's what it means is, we're using similar structures that we know and their functions and kind of inferring or implying it to these unknown proteins. So a motif is actually a reoccurring thematic element in music or design. Design, that's what I'm talking about here in cells or proteins. So like one example of a motif, in every James Bond movie you've ever seen, they say shaken, not stirred. He orders a martini. I mean, you expect that line. In every Star Wars movie, I should add this, they say, I have a bad feeling about this. It's there. It's very subtle in some scenes, but it's there. Movie directors have their own little motif, if you will. Michael Bay movies always have massive explosions. Transformers, that's just one. For some reason, Michael Bay, all I think about is Transformers, but Michael Bay has a lot more movies out there. Christopher Nolan, the director of Inception and Interstellar, he likes to mess with your mind, okay? And then Alfred Hitchcock usually does something with a triangle with his characters. There's always kind of like a triangle, three different main characters, and they kind of interact. So that's kind of an interesting one if you're into movies. Look up Alfred Hitchcock and the triangle. Okay. But motifs, proteins have motifs as well, and so there are three motifs that I'd like to discuss with you. The beta, alpha, beta motif, how it creates a fold or a crease. Beta, alpha, beta motif is also known as the Rossmann fold. So here is the beta, alpha, beta. So what happens is you get the paper fan shape, then it turns into a helix, then it goes back into a paper fan shape. So that's all it's saying, beta, alpha, beta. Beta barrel, beta sheet folded around to form a tube. So you got the paper fan, and then you just roll it into a cylinder. So aquaporins are beta barrel. Aquaporins allow water to enter and leave the cell very rapidly. So we'll talk about aquaporins and their use with like freezing of cells. And then this is a big one, the helix turn helix. That one is listed here. helix, turn, helix. This is super important because proteins, there are proteins that just bind to beta, turn, helix. What did I just say? Beta, turn? Oh my gosh, I meant to say helix, turn, helix to the DNA. So proteins recognize the helix, turn, helix. They bind to it, maybe turn it on a gene or turn it off a gene. So those are the three motifs that we use with proteins. But there's a lot more, but these are just the three big ones. We also use motifs in the analysis of proteins. Kind of helps shed light of unknown functions or maybe unknown origins. So we can compare motifs found in different organisms and kind of look at how closely related they are.
Can I have your attention, please? There will be no junior high football game in Ross Bay today.
Okay, that's nice to know. I'm going to cross that off on my board.
You will have practice.
Oh, that's a bummer.
Again, no junior high
All right,
football game.
see you later, Herman
You will have
Norcross.
practice.
All right, domains, functional units within a larger structure. So we have a substructure within a tertiary. So here's an example because that probably doesn't mean anything. Your amino acids, think of them as letters. Your motifs are words or phrases, and the domain would be the paragraph, okay, that results from all of this. Most proteins are made up of multiple domains, can be physically separated, and it also deals with the folding of the protein into its proper shape. So this is where I'm going to segue into chaperones. But here's a picture of domain just to give you an idea. Here's a motif. I have my beta, alpha, beta. So I think it starts here with beta. Nope. That doesn't look like a beta, alpha, beta to me, but whatever. Okay, so it's beta, alpha, beta. I think they messed up on their picture. But it's a portion of this huge globular protein, so there's that domain. Yeah, that picture's not right. That one is actually like an alpha, beta, alpha. No, wait. No, it is right. Beta, alpha, beta. Okay, never mind. I just looked at it wrong. So folding of proteins rely on something called chaperone proteins, so proteins that help other proteins fold correctly. At first, we didn't know how proteins folded. We just thought they did it spontaneously due to their hydrophobic interactions with water and other nonpolar amino acids. Proteins can fold in so many ways. So it's like, how did the protein know? You know, does it just do trial and error? Nope, can't carry oxygen. Unfold and refold again. Oh, now I can carry, like, what's the, there's got to be something in charge here. So it took way too long. So how do proteins fold? Well, they noticed that there was an unusual mutation in bacterial cells that prevented viruses from replicating. So then they found that the normal bacterial cells had chaperone proteins, and the ones that were mutated did not have chaperone proteins. As a result, couldn't make more viruses. So I don't know if you know what viruses are, but viruses are considered non-living because they cannot reproduce on their own. So I always like, this is a horrible analogy, and I'm sorry for being so blunt and, I don't know, whatever other word to describe it. But I always like to think of viruses as terrorists. They hijack the cell. They say, hey, make more of me or I kill you. So the cell's like, okay, makes more of the viruses. Viruses explode out of the cell. You die anyways. Okay, so terrorists, they hijack cellular mechanisms to make more of themselves. But these mutated bacteria cells didn't have any chaperones. Chaperone proteins. So these viruses, okay, so we made all these viral parts, but we couldn't assemble them properly. So then viruses, you lose. So.
Where it says explain the role of chaperone proteins and protein folding, sorry, just write terrorist.
I would say that it just makes.
No, I'm just kidding.
I would honestly just say that it just makes sure that the proteins fold correctly, like they do it in the correct way. So this diagram down here kind of shows what a chaperone protein does. So we have a chaperone protein, and this is a misfolded protein that needs a little help. So then the chaperone protein takes it in, helps fold it correctly. It kind of looks like they put a cap on and incubates, whatever, and then release. You're folded properly. Go out into the world and do your job. So, chaperone proteins help other proteins fold correctly. There are many proteins that act as molecular chaperones. So, just because I'm saying chaperone proteins, there's not just one type. There are many types. During heat shock, which means you increase the temperature, proteins become unfolded. And you can refold those proteins by, adding chaperone proteins or having them come in contact with each other. So here we have a chaperone. I know this is kind of a cheesy diagram, and this unmisfolded protein. And what the chaperone protein does is it refolds it and then creates what it should have folded into. So they call this the native protein. Now, some diseases result from improper folding. So a disease caused by chaperone protein deficiency. So that means that your chaperone proteins don't work right. That's what it means. Cystic fibrosis, vital protein involved with moving ions across cell membranes. So this picture right here is showing cystic fibrosis. Here's the cell membrane, outside the cell, inside the cell. And in order to keep your airways clear from mucus, because mucus is used to trap dust and bacteria, you use chloride ions, okay? So then in a normal CFTR channel, because that's where the deficiency takes place, it moves chloride ions to the outside of the cell. So then these chloride ions go out, and then the mucus is cleared. It runs perfect. When you have a mutated CFTR channel, because maybe the chaperone proteins can't help fold it correctly, you can't move those ions outside the cell, and as a result, mucus builds up, and then basically suffocate on your own mucus. So it's just a horrible disease, and it's all because of a faulty chaperone protein. Alzheimer's, your book mentions Alzheimer's. Alzheimer's is still being investigated as to how does it happen. I feel like every day we learn something new about Alzheimer's, but I'll just tell you what your book stated. Protein clumping in brain cells produces these amyloid plaques. So proteins build up, and whatever chaperone is supposed to refold or kind of get rid of it, well, there's a deficiency there. But we do know that if you have Alzheimer's, we do find amyloid plaques, and so that's just one of the hypotheses as to how does Alzheimer's develop. Do you have some faulty proteins in your brain that are not working correctly, and it just builds up over time? Who knows? Okay, denaturation inactivates proteins. So if the environment of the protein alters, maybe you're going to increase temperature or decrease the temperature. Or you're going to change the pH or ionic concentration, salty, not salty. The protein shape can change or unfold. And so in your lab, you will subject protein to different temperatures. There's a part in there where you're going to use a hot water bath versus room temperature. There's a part in there where you're going to figure out which pH is optimal. So you're going to use pH of 1, 3, 5, 7, so on and so forth. And you're going to see what happens to the protein. So when these proteins become denatured due to their environment change, they are biologically inactive. They don't work anymore. So pickling and salt curing are used to keep microorganisms from growth or damaging your food.
Can I have your attention for the announcement?
Because you are destroying their proteins.
Seniors, if you'd like to submit an application.
This is
for
the last slide, so I'll just read this. Enzymes have a narrow range of physical parameters, so they have an optimal pH or temperature that they work best at before they become faulty. So I'll pick it up here tomorrow.
See Mrs. Young on SR if you have questions.